As many of you know, HS is a chronic inflammatory skin disease, and it occurs when you have a hair follicle that becomes occluded and it dilates, and eventually it ruptures. When it ruptures, it incites an inflammatory response, and you get the recruitment of a variety of immune cells, as well as the secretion of pro-inflammatory cytokines. Eventually, this can progress to a chronic state where you get the formation of sinus tracts and scarring in the lesion as well. And we really do think that the pathogenesis of HS is a multifactorial event that causes these changes in the tissue. So we know genetics and mechanical stress play a role in HS. We also think that there's a contribution from the microbiome being dysbiotic, as well as these aberrant immune responses, and this can contribute to potentially changes in the skin structure and function. However, there's still a lot that we don't know about the inflammatory mechanisms behind HS, particularly what the mechanistic differences may be between the Hurley stages. So in Hurley stage 1, we get the formation of these nodules. In Hurley stage 2, we get those sinus tracts forming, and in stage 3, you get that extensive scarring and a much broader lesional site. And particularly, we want to understand not just the differences in the Hurley stage, but what the epidermis is doing in each of these stages. Because we do know that the epidermis is hyperplastic in HS, and we have studied the dermal component of the inflammation quite extensively, but we know very little about why this structure comes about and what particularly is going on with the inflammation in the epidermis specifically. So in our lab, we get samples from the dermatology clinic and plastic surgery, and we're able to use these samples for a variety of downstream assays, including histology. We can also take the tissue and dissociate the dermis and epidermis for further downstream assays. And we can culture keratinocytes as well from the epidermis and do assays on these cultured cells as well. But to start answering the question about the contribution of epidermal inflammatory activity, the first thing that we wanted to do was take some blinded H&E samples from healthy control patients, non-lesional skin from HS patients, as well as skin from all 3 of the different HS lesional stages. And we had a dermatopathologist rank the epidermal and dermal inflammation separately on these samples, as well as help us to start identifying some of the immune cell types present by the location, either epidermis or dermis. And then we took the ranking for this and unblinded it and compared it to the clinical staging. Now we have a lot of this in graphical format, but for time, I'm just going to show you a summary of what we found. So with a healthy control and the non-lesional samples, we see that inflammation in both the epidermis and dermis is minimal. You get some infiltration of immune cells, but not too much. They're quite similar. But when we start to separate out the lesional stages in the epidermis and the dermis, we start to see some differences by stage. Particularly with the epidermis, we see the stepwise increase in the level of inflammation by Hurley stage. So you see some inflammatory cells, but it's minimal in stage 1 epidermis. This increases in stage 2, and then you start to see a lot in the epidermis by Hurley stage 3. However, with the dermis, you see a more constant level of inflammation occurring with a decent amount of immune cell infiltrate, even at Hurley stage 1. We were pretty curious about how this response in the epidermis is evolving and how the immune cells are increasing by stage here and what those immune cells might be. So to answer the first question about what types of immune cells are there, so I'm showing you Hurley stage 3 specifically here, but we have looked at earlier Hurley stages as well, but this is when the majority of the cells are there. You get CD8 T cells in the epidermis, NK cells, as well as neutrophils. So CD8 T cells and NK cells are also present in earlier lesions, generally at a reduced number. Neutrophils are pretty specifically found in Hurley stage 3 lesions, so only the most severe inflammation is really including neutrophil presence in the epidermis. We wanted to understand how these cell types are recruited to the epidermis, and our first hypothesis was really that keratinocytes may be the recruiters of these cells. And so we assessed the chemokine expression from keratinocytes in healthy control samples, as well as stage 1, 2, and 3 lesional keratinocytes. And what we found is, in fact, that these keratinocytes secrete chemokines that recruit neutrophils, such as CXCL3 and IL-8, as well as T-cell and NK-associated chemokines or broader action chemokines that can also recruit T-cells to the lesion, such as CCL3, CCL2, and CCL22. These chemokines do kind of vary in expression by stage, and this very well matches when we're seeing the infiltration of the specific immune cell subsets into the epidermis, indicating that keratinocytes do likely provide a stimulus to get these cells into the epidermis. So the next question we had was, what cells are really responsible for the inflammatory cytokine secretion in the epidermis? And keratinocytes are one of the major contributors here of specific pro-inflammatory cytokines. A big one that you see here is TNF-alpha, for which adalimumab targets in HS therapy. So we see that particularly in all three lesional stages with TNF-alpha, we get a lot of of HS, we get a lot of TNF-alpha production from specifically keratinocytes. And this is pretty drastic, even in comparison to the dermis. So this is pretty important. IL-6, we also see a similar pattern where we're getting the keratinocytes being the primary expressor of this particular pro-inflammatory cytokine. So keratinocytes are responsible for some of the pro-inflammatory cytokine secretion to a large extent in HS epidermis. But we also do have a contribution from immune cells as well as immune cells particularly triggering keratinocytes to further produce additional cytokines. So this communication is important and allows for the production of increasing levels of interferon gamma with Hurley stage in the epidermis, as well as IL-17. We think that these are coming from, particularly gamma is coming from T cells or NK cells. And IL-1 beta also increases, and that's being produced by both immune cells and keratinocytes in our HS lesions in the epidermis. And then also type 1 interferon production is increased as well compared to healthy control. So kind of summarizing some of this data, a lot of the current therapies out there, the approach is really to target individual cytokines, but here even in the epidermis, we have kind of this evolution of cytokine responses in the different Hurley stages of the lesion. And really targeting one cytokine at a time here may not be effective necessarily in HS therapy, as we see that some of these therapies only effective in maybe 20 or 30% of our HS patients. So we were wondering if potentially other therapies that target cytokine secretion more broadly may be more effective. And that brings us to the JAK-STAT signaling pathway here. So JAK-STAT signaling could be potentially a topical drug target based on our data, and I'm going to get into that in a minute. But JAK-STAT signaling has been targeted in other dermatological conditions, which are outlined here. So we already know that it can be effective for other dermatological conditions, and some early trials of oral Ruxolitinib have shown success in HS patients, but they do come with side effects. So potentially a topical could be a better option in light of the side effects of a systemic therapy like that. So kind of starting to test the effect of Ruxolitinib in some of our cells. First of all, we wanted to confirm that STATs are expressed more highly in HS. So we assessed STAT1 and STAT3 expression in healthy control epidermis versus HS epidermis, and did in fact find that there is increased expression of the STAT proteins. And then what we did was we took lesional keratinocytes from stage 2 or stage 3 lesions, and we cultured them and treated them with either vehicle control or Ruxolitinib in culture. And treatment with Ruxolitinib was able to decrease a variety of key cytokines and chemokines about twofold, while it didn't touch some of the other ones involved. So it does have a broader but still somewhat specific effect on inflammatory cytokine and chemokine secretion. So we thought that this is pretty promising for HS therapy potentially, especially the implementation of a topical therapy that can target inflammatory activity more broadly. So just to summarize this, kind of what we found here. So We do find that there is an evolving inflammatory response in HS epidermis by Hurley stage. In the most severe cases, you have a variety of immune cells present, including NK cells, neutrophils, and CD8 T cells. We think that those are being recruited by keratinocytes through the production of a variety of chemokines. And what we find here is that Ruxolitinib may be able to effectively block both the production of the inflammatory cytokines in the epidermis as well as the further recruitment of immune cells. So it's acting in several different manners here to potentially be blunting inflammation. And this lends itself to potentially a topical therapeutic for either Ruxolitinib or a drug with a similar targeting profile and potentially efficacy.